Hello everybody. So today I'm basically going to show you how a neuron works. In case you don't know what a neuron is, it's one of the little cells in your brain that helps you perform basically anything. Okay? So I'm going to give you an overview of all these parts first, and then I'm going to go into a little more detail as a process. So we have our receptor for our dendrite. So receptor, dendrite, nucleus, cell body, axon hillock. This entire tube here is the axon. The little brown parts are the nodes of Ranvier. The blue is the myelin sheath. This right here is our autoreceptor. Here's our terminal branches that end in terminal buttons that hold our vesicles. See, terminal buttons. Reuptake channel, enzyme, receptors, and here's another cell. And that's going to receive the signal this cell puts out. Okay, so over here is basically this is kind of like the inside of the cell body. We have our proteins, which are negatively charged, our potassium, which is positively charged, our chloride, which is negatively charged, and our sodium, which is trying to run away, and that's positively charged. This right here is a membrane. This is our potassium channel, sodium potassium pump. This is the inside of the cell, and this here is the outside. We'll come back to this later. Alright, so first, our cell's going to receive a neurotransmitter. And this neurotransmitter gives our cell the signal to go ahead and work out this process that I'm going to explain to you. So, here's this, and this step's called initiation, which is just what I just said. Neurotransmitter hits the receptor of the dendrite. Okay? And the next step is called transduction. And this means that the chemical signal of our neurotransmitter is going to be changed into an electrical one. And right now our cell's in resting potential. Resting potential means it's not doing anything, it's just kind of waiting for it to be told what to do. Waiting for a chance to fire. It's along those lines. Okay. And we're going to move over here again. So right now, our potassium channel is open, which means our potassium ions have the freedom to move in and out of the cell. They can do whatever they want. Our sodium ions are stuck here because their little channel's closed. And our potassium is going to move in and out because of diffusion force and concentration gradient. So they spread out, and the concentration differs. Like how many there are differs, but you don't really need to know that. You just need to know like they spread out because they don't want to be next to those with the same charge. Okay. So when this signal from our neurotransmitter hits this part of the cell, our gate's going to close, and this straw is rebellious and it does not want to close. But go ahead and pretend that's closed. Use your imaginations because this one's taped down and I can't replace it. And our sodium channels are going to open. And then when this happens, our sodium ions are going to go, hey, this is awesome. We're going to flood inside here. And the reason they get in there, dude. And the reason they do this is because of ionic force and diffusion. So, diffusion force. So, the ionic force has to do with the fact that inside here, it's negative 70 millivolts. That means the inside of the cell is just all negative. And these are positive. So, opposites attract. They're going to be attracted to the negative charge inside the cell. And the diffusion thing says that they don't want to be next to each other because they're all the same charge, so they're going to spread out. They say, hey, I don't want to be like you. I'm just going to be separate from you. I'm just going to be spread out all over the place. Okay, so when these all flood inside, it changes their charge inside the cell. It goes from negative 70 millivolts to positive 40 millivolts. And when this happens, our sodium gate's going to close, trapping all our sodium inside, and our potassium gate's going to open. And our potassium says, well, we don't want to be in here. It's too positive. So they're going to fly out into the outside of the cell. Okay? So they're going to go out into the outside of the cell. And this right here is called, and this entire process of the sodium coming into the cell was called action potentials, taking action and carrying out the electric field. That's a way you can remember it. This here is... This is refractory period 1. Okay, so how'd that get in there? Get out, dude. So, we're working on reestablishing kind of what we had before. Alright? Alright, so now we're back to negative 70 millivolts. Okay, and this causes our little potassium channel to close again. Okay. Okay, so this is great, but notice something different? All our guys are on the wrong side of their membrane. Now the charge is the same, but our cell can't fire anymore. So it's got to reestablish that previous state it was in. This is like refractory. It's called refractory period 2. So remember our sodium-potassium pump? It's going to use energy to exchange these ions, exchange these ions so that it can fire again. So it's going to exchange all the ions, and I'm just going to go ahead and toss these, but know that it usually takes 1 sodium ion per 1 potassium ion. But for time's sake, we'll just toss them all out there. Okay? So this process right here is what's occurring all inside the cell body. It builds up an electric signal. It builds up the charge. So I'm going to move back over here, and I'm going to go into step 3, which is propagation of the electric signal to the axon hillock. And all this charge is building up and is moving to the axon hillock, which ultimately decides whether or not the signal is going to continue down the axon. So if it reaches negative 55 millivolts, like the combined charge of all our little guys, then it's going to fire. This is step 5, whether or not it's going to fire. And then we're going to move on to step 6, which is saltatory conduction. Okay, so now outside this axon, there's a bunch of sodium ions too. They're just kind of all over the place outside this neuron. And when the signal reaches the axon, assuming that we're allowed to fire, all these sodium channels along the axon are going to pop open on us and let in all the sodium. Okay, so here's our sodium ions, and they're going to pop inside the cell. So we're going to go. This would go much faster in actuality, but I'm not that fast. So I'm going to pop them inside our axon, and this is going to continue all the way down the cell, as you can see, all the way down the axon. Okay, the inside of the axon is negative because it contains some proteins that are negatively charged. So the sodium's attracted to that. So that's the reason it's going to flood in, because of its ionic force. And this myelin sheath right here is going to insulate our charge. So our sodium's going to stay positive, and our protein's going to stay negative. And this causes it to shoot straight down the cell. It just goes so fast. And it's so fast that it doesn't even look like it's moving. It looks like it's jumping from node to node. I had a light to show this, but you can't really see it. So I'm just going to demonstrate using my finger. It goes all the way down to the terminal branches. And when we get to the terminal branches, which contain our terminal buttons that hold our vesicles, the signal causes our vesicles to spit out their neurotransmitters into the synapse, which is this gap right here. It's a space between neurons, basically. Okay? And it's going to spit them out into this gap. And this step here is called transduction back to chemical. So we're going from our electrical signal that was inside the axon to a chemical signal by releasing the neurotransmitters, which are chemicals. They're not electrical in any way. They're just influenced by electrical signal. Okay? And then they're converted back to electron when we get to the next cell, but we've already covered that. Okay? So now they're all inside the synapse. And at this point, 4 different things could happen, which is step 7. It dictates what happens next. Either it could be taken up by our receptors on the next cell, which signal the cell over here to continue the process. So it's going to gobble them up and say, hey, we can move now. We can do exactly what this cell just did. It's going to hold those. And it doesn't want to close. Okay, dude, close. Okay, you're probably a year older now since I got that closed, but there you go. Or, see, we have neurotransmitters all on our synapse, and we do not want this. We want to clean it out. Okay? So it could be sucked into the reuptake channel and recycled into another vesicle. Our neuron here doesn't want to waste any energy. It's going to dedicate all its energy to doing stuff it needs to do. And if a neuron's hanging out in here, it doesn't even make more. It could just use the ones it has. So it's going to use that energy to reuptake our little neurotransmitters into the vesicles. I'm going to take this back out again. But you get the idea. It's reuptaken. I just need it for another thing. Or it could go to our autoreceptor right here. Okay, once it reaches the autoreceptor is going to signal, hey, stop to the neuron. And that's going to escape, okay? It's going to signal, hey, stop to the neuron. It's going to say, we don't need to keep going. Our signal's done. We don't need to produce anymore. You're done working. It's time to stop work. That's what it basically tells it. Or the inside could come along in here like a little vacuum cleaner and eat up the remaining neurotransmitters. And it just cleans up the synapse. It cleans up all those neurotransmitters we have laying around. It's hanging out in the synapse waiting to clean. Alright, and then this process continues down the next neuron, and then the next, and then the next, and you get the idea. And that's about it. Okay? So I hope you learned something, and that's how Neuron works.